Hello, everyone. Welcome to join today's MIT Chief, the Future Lecture Series. My name is Yong Shen, and I'm a postdoc at Harvard Medical School. I also serve as co-president at MIT Chief. MIT Chief's full name is MIT China Innovation and Entrepreneurship for RU, which is committed to promoting collaborations between China and the United States on innovation and entrepreneurship. This year, we launched a new initiative event, the Future Lecture Series, in which we invite leading scientists to discuss breakthrough technologies moving humanity forward greatly. Through the series, we hope we can connect our audience with those pioneer scientists and entrepreneurs, hearing their exciting findings and insights, and envisioning how our future will be shaped. Most importantly, we hope this lecture series could inspire people to rethink the future, explore the future, and meet the future. Next, I would like to introduce our moderator for today's lecture, Audrey Lee. Audrey is a sophomore student at MIT, major in computer science and economics. So let us welcome Audrey to moderate this fantastic lecture.
Thank you, Yong. Hi everyone, thank you for joining us today for the MIT Chief Future Lecture Series, Episode 2, Next Breakthrough in Neuroscience. Obviously, 2020 is not an easy year from any aspect. So by launching this series on this virtual platform, we hope we can contribute and continue to create value for all of you who are curious to learn and passionate to make a change. Given that most of you have already seen and read our event flyer, I'm here with Professor Robert Desimone. Professor Desimone is the director of the McGovern Institute and Doris Don Berkeley Professor in the Department of Brain and Cognitive Sciences. Prior to joining the McGovern Institute in 2004, he was the director of the Intramural Research Program at the National Institute of Mental Health, the largest mental health research center in the world. In today's talk, Professor Desimone will share with us his findings and insights around novel therapeutics for treating brain disorders, gene therapies, direct manipulation of brain circuits, and precision pharmacological approaches. During the talk, please feel free to click on the Q&A button at the bottom of the Zoom window and submit your questions in the box. In the meantime, you're welcome to like anyone else's questions that interest you. After Professor Desimone's keynote speech for about 40 minutes, we'll initiate our Q&A session and select questions with the most likes. And without further ado, please join me in welcoming Professor Desimone to begin his keynote speech. Professor, if you would like to screen share. Oh, I think you're muted, Professor.
Okay, can you see that?
Yep.
Okay, very good. Well, first of all, I want to thank Chief for inviting me to give this presentation tonight. Your goals are shared with me, I can tell you. And I should also tell you that I'm speaking from my virtual office at MIT, not my real office. I'm actually in my house because of the limitations of COVID. But be that as it may, I will start with telling you a little bit about the motivation for us to do this work. And the McGovern Institute was started about 20 years ago by Pat and Laurie McGovern with a dual mission, which was to understand the brain and then to apply that knowledge to help people with brain disorders. And tonight I'm going to be focusing on that latter part of the mission. How can we actually help people? And the need is really urgent. If you look at the prevalence of many of the major brain disorders, there's just millions and millions of people affected. And the really bad news is, if you look at the prevalence over years, the incidence of these disorders is actually growing larger, not smaller. We're not curing these disorders. In fact, we're not even really treating them very well. The treatments that we have for brain disorders have really not improved very much over the past decades. Most of the medicines that we're using were discovered serendipitously. Now we just try to find other drugs that look like the older drugs. And the most important thing is they're not based on an understanding of the underlying mechanism of the disorder. And that really is in contrast to some other areas of medicine where there's real progress that's being made in the last few years. And an obvious example is an infectious disease. Take HIV, for example. Once we began to understand how HIV ruins our immune system, it was possible to develop the antiviral drugs, which have now had a dramatic impact on deaths from HIV. It's one of the great success stories, and I hope someday in the not distant future, I can look back and tell you about the great success story that we've had with COVID-19. Another area of medicine where we've had these sort of mechanistic success stories, you can see here from the incidence of deaths from cancer across all areas of the country over years. And you can see that deaths from cancer are also falling. And again, this is coming about in large part through these targeted mechanistic treatments. And I'll just give you an example from one of the targeted treatments, which is the treatment for chronic myelogenous leukemia, a cancer, which was treated with Gleevec. And some years ago, it was discovered that there was a chromosome break in 2 fused chromosomes, and then a fusion gene that resulted in a novel protein. And that novel protein, unfortunately, caused the uncontrolled cell proliferation. But once that was determined in the 1990s, that protein then was the cause of the leukemia, chemists could step in and find molecules to block the action of that protein, bind to it. That turned out to be Gleevec. And by 2001, Gleevec was released as a drug. So great success story. So this is what we need in the whole field of brain disorders. Now, there has been a recent example of a mechanistic breakthrough in the field of brain disorders, which is just in the last few years, the treatment of spinal muscular atrophy. And here it was discovered that the spinal muscular atrophy results from a mutation in the SMN1 gene, which results in a degraded protein. And as a result, you have great disability and even death due to the loss of motor neurons. It was also discovered that—
Sorry to interrupt, Professor. Do you mind switching off the presenter view? I think it's currently in presentation mode. One of the audience members said they couldn't see the slides very clearly because it was like it shows this slide, both this slide and next slide in the series.
Oh dear. Okay, I am not entirely sure. Hang on. Is this better?
Yes. Yeah, this would be better. Or let me find the presenting. There might be one that allows yeah, maybe try clicking on that one. See if that would.
Yeah, let me try something else. Oh no, let me see. Oh no, let me try something else. I'm just try sharing the screen differently. No, that's not going to help me. One second. Okay, now let me try. Okay, and now let me try the screen share again. But I'm going to choose a different screen. Okay, how about this? Is that better?
Let me see.
I can't see what you're saying.
Yeah, this is perfect. Thank you so much.
Okay,
Sorry about the interruption.
No problem. It's just that now it's for me, it's over here, but that's no problem. 1 second here. Okay. So anyhow, so it was also, yeah, unfortunately, I got to look over here. I guess I hope that's okay. It was also discovered that there's another gene, related gene, the SMN2 gene, that makes a little bit of functional protein, but not a lot. And so there was an effort then to develop an antisense RNA that would correct the splicing of this gene and resulting in now a much greater quantity, but now I don't know how to change the slide. There we go. A much greater quantity of the functional gene. And even more recently, there's been another approach in gene therapy, which is actually just to use a virus to insert the functional gene, and that again can make more of the functional protein. So here we have, again, sort of a mechanistic understanding of the disease, and that's led to a really important treatment. So how can we get to this mechanistic understanding of other brain disorders? So I'm going to tell you a little bit about the McGovern approach, and then give you some examples of, I think, what's going to get us there. So in order to understand how we go from a gene mutation or altered circuit to a complex behavioral phenotype and altered emotions and memory and so on, we really need to understand the system at multiple levels, going from the cell molecular level all the way up through the computational level. And so we need to recruit faculty members that span all those different levels, which is what we've done. As you can see here, the faculty members go from cell molecular, cognitive, systems, and computational, and many of the faculty members sort of bridge levels, so we can communicate across all these levels. And then the other major aspect of our approach is international. And this really began at the start of the McGovern Institute, where, again, Pat and Lori McGovern believed that we're working on global problems, and we need, therefore, global solutions. And because of their particular interest in China, they established the 3 IDG McGovern Institutes in Beijing, at Beijing Normal University, Tsinghua University, and Beida. And then in recent years, we set up a major collaborative program with SciAT, the Shenzhen Institutes of Advanced Technology, and I'll be telling you more about some of those collaborations. And the idea is that by working together across the US and China and other international countries, we can accelerate the pace of discovery and accelerate the pace of new treatments. And again, I'll give you some examples of this. I should also say that the Chinese institutions we work with have adapted the same multi-level approach that we have. They have people spanning from cell molecular through systems, cognitive, and up through computational models of brain function. So we're all rowing in the same direction. All right, so now let's talk about how we're going to get to these new therapeutics. Well, what I'm going to try to convince you of tonight is that new tools and technologies are driving this development. They're driving an enhanced understanding of neural circuits. They're leading to better animal models. They're leading to a better understanding of the genetic basis of disorders and better brain imaging methods that are all moving us in the direction of better treatments. Now, one of these breakthrough technologies that has really revolutionized neuroscience is known as optogenetics. And optogenetics was co-developed by Ed Boyden, one of our faculty members, when he was at Stanford. And it depends on using a virus to insert genetic material into cells, which makes those cells sensitive to light. And then, using lasers, you can stimulate those cells and turn them on or off, depending on the options you use and so on, and basically play them like a piano. And so, as you may imagine, this is a tremendous research tool for probing the functions of neural circuits. But as I'm going to show you later, it could potentially also be an important therapy. Now, I'm going to give you an example of how we're using optogenetics to probe circuit function and as a potential therapy through the work of Fan Wang. Fan Wang is currently on the faculty at the Duke University, but we've recruited her to the McGovern Institute, and she'll be starting in January. And Fan has recently started the study of pain. Now, pain is a huge area of public health impact. It's a big problem in the US, chronic pain. And because of chronic pain, there's been this overprescribing of opioids. And now we have an epidemic of opioid abuse and opioid addiction. It's just a terrible society phenomenon. And so what we desperately need are better ways to control chronic pain without the use of opioids. So to help get us there, what Fan did was she injected mice with a whole variety of different anesthetics, work in different ways. And what she found is even though they have different mechanisms of action, they all stimulated a particular group of neurons in the central nucleus of the amygdala. And you can see here, they look like that little field of stars there. And this is a blow up, so you see them all in the different colors. And they were the common denominator for all these different anesthetics. Then based on this and other knowledge she has about the system, Fan hypothesized that in chronic pain, what happens is these neurons are understimulated. And what the anesthetics do is stimulate them. These are inhibitory neurons, I should add. And what the anesthetics do is stimulate them. And that turns off activity in a whole variety of brain structures that are more involved in the perception of pain and all the emotional consequences of that, the emotional cognitive consequences and so on. So her idea was if she could stimulate these cells, and in this case with optogenetics, then if her hypothesis is correct, then this should result in the relief of pain. So she took mice that had a chronic neuropathic pain in their face, and she injected this optogenetic construct so that it was taken up by these cells in this nucleus. And then without the light on, she stimulated the face of the mouse with the probe, and they reacted right away, like as it was painful. But then she turns on the light, which is a fiber connected into the animal's head. And then when she probes with the same calibrated probe on the face, the animals don't react at all. They couldn't care less. So she had cured the chronic pain in these animals. Now, obviously, this would be huge if this would work in people. So how could we do it? Now, the one way could be if we could find a drug, for example, that just targeted these cells. Difficult, but something that is being worked on. But other possibilities are to stimulate the cells with other means. And for example, maybe with optogenetics. Now, there's a practical difficulty here because the human brain is a lot larger than the mouse brain. And the light that might only have to shine a short distance through a mouse brain to stimulate these cells might have to travel a long distance through the human brain. And light doesn't travel very well through brain matter. It gets absorbed, and so not much reaches the cells. So what Guoping Feng did, and he's another faculty member in our institute, was he developed an opsin that was much more light sensitive than the opsins that we were using to that time. It was called SOL. And not only is it much more sensitive to light, but it also acts as an on-off switch for neural activity. And what you're seeing here is a firing rate histogram. And what he found was when you stimulate the cells with blue light, it turns the firing on and it stays on, like you flip the lights in the room. And it turns off when you shine orange light. So he had a way of controlling activity for long periods. And he confirmed in primate studies, which we've now published, that it works in primates and it works over many millimeters of brain matter. Although it does still require that it requires some light source on the head, and a hole through the bone. And so this is, potentially an optogenetic therapy, the basis for a therapy. Now, we have another faculty member, Polina Anakiva, who's working on implant-free, let's say, stimulation methods based on magnetic fields. Now, magnetic fields don't get absorbed by the brain. They just pass right through the brain. So there's no limitation there. So what she does is she injects magnetic nanoparticles into the neural population that she wants to excite. And then once she's injected these nanoparticles, she can stimulate with pulses of these magnetic fields and activating the cells. And what you're going to see on the next slide is an image of calcium imaging of mouse neurons when she has stimulated them with the magnetic field. You see them here, they're those white blobs, and when you see it on, now you can see that the field of neurons has become activated by that magnetic pulse. So that's another promising avenue for being able to target neural populations in the future once we've identified the relevant target. Polina is also developing something that she calls chemomagnets, which is really another fascinating idea, these are liposomes loaded with magnetic nanoparticles, which may allow for multiple cycles of drug release. So in these liposomes, you have the magnetic particles, you have drug, and she can inject these into precise areas of the brain. And then she applies the magnetic field, and then you can very precisely deliver a drug or a medicine to a small neural population. And this is instead of standard pharmaceuticals where you take a pill and it bathes your whole body and whatever that drug is with all the consequences. So again, these are, new technologies which are then sort of leading the path to what I believe could be the new therapies of the future. Now I want to turn back to the genetic basis of brain disorders. Over the last couple decades, we've had an ever-increasing number of what are called genome-wide association studies, or studies that have tested large numbers of people, many thousands, tens of thousands, to look for associations between genetic mutations and vulnerability to developing a brain disorder. And these studies have identified many of these mutations or polymorphisms that lead to this vulnerability in all sorts of disorders, hundreds of them related to bipolar disorder, schizophrenia, depression, and so on. And now that we are accumulating this genetic knowledge of the genetic basis for brain disorders, we're now beginning, some of the pieces are beginning to become clearer. And one of the things that we are learning from these studies is that the single diagnostic categories that we use, whether it's bipolar, autism, whatever, in fact, could encompass multiple different genetic mechanisms. So in autism, for example, it's a very heterogeneous disorder. You have some people very, very high functioning, super functioning, and you have some people that are really severely intellectually impaired. That there are different subtypes with a different genetic basis. Now, if we look at what some of those genes are that have been identified that relate to these subtypes, this shows here a timeline of gene discovery in which you can see the individual genes that have been identified. Many of these genes, I should say, also, they're of large effect. So for some of these, if you have this particular mutation, you're at high risk for developing this form of autism. Now, one of the other things that we've learned, now that we're accumulating this genetic knowledge, is that in autism, many of these genes express proteins in the synapse. Now, the synapse is where the neurons are communicating with each other. This is, like the rubber hits the road, at the synapse in the brain. And so this is, could potentially have a very big impact if you're affecting those proteins. This just shows a diagram of some of these proteins that are affected by these autism-related genes. And it turns out one of these proteins is known as SHANK3. And it's the mutation in the SHANK3 gene leads to a particular subtype of autism known as Phelan-McDermid syndrome. Now, Phelan-McDermid syndrome represents about 1% of the cases in autism, but it actually represents a much higher percentage of the cases that have the severe intellectual disability. Now, if we go to what are some of the impairments that you see in Phelan-McDermid syndrome, they include the social impairments common in autism, intellectual disability, and many somatic disorders. Some have epilepsy, speech may be delayed or even blocked altogether. They have difficulty sleeping, and many more disorders that I don't have here. So how do we go from this genetic mutation to an understanding of how the gene leads to these autistic symptoms? And even more importantly, how can we develop a treatment for it? Sort of like we saw for SMA, for example. So here we go to another really breakthrough technology that's giving us this capability, and that's CRISPR. Many of you may have heard of CRISPR gene editing, which was invented in cells by Feng Zhang, another one of our faculty members. And CRISPR can be used to edit the genome, correct mistakes in the genome, and it has been used in animals and unfortunately in a human case to do genome editing in the embryo. And we're definitely not following that here at MIT. But it could also be used to create animal models with these mutations, and could also, variations of this could be used even as a therapy. So it's a research tool and potential therapy. So how do we use this for SHANK3? Well, you can use CRISPR to introduce this mutation into animals. And this was done by Guoping Feng and then other labs around the world, creating mice with the SHANK3 mutation. And the mice have impairments that parallel some of the things that we see in human autism, and mouse kind of versions of them. Which tells us maybe we're on the right track. But the problem with these studies in mice has been that although they tell us a great deal about the biology, they're incredibly important to understand what's going on in the brain, that when it comes to treatments developed in mice often do not translate into human clinical success. And this has been the subject of numerous articles, papers, news articles, and so on. How is it that something that cured a mouse doesn't cure a person when it's tried? And in a disorder like autism, perhaps it's not too surprising. I mean, it's affecting the brain, and the mouse brain is obviously much smaller and has different characteristics than the human brain. And particularly in autism, we know that, for example, social cognition is a function of activity in a structure known as the medial prefrontal cortex. And then you can see that here in this colored region over here. In humans, this is really big and really elaborated. In mice, it's very small and much more primitive. However, in monkeys, which are non-human primates, it's actually much closer to the human structure. And because in many ways, the monkeys and humans were all primates, and we share many characteristics. This is a reason for us to believe that maybe if we had a monkey model for the SHANK3 mutation, and then we developed a treatment in a monkey, then that would more likely translate into human success. So what we did was we formed a collaboration with the Shenzhen Institute of Advanced Technology, which I had told you about previously. Now, in Shenzhen, they had access to large numbers of monkeys and had a lot of experience with a large animal in vitro fertilization. They had a great neuroscience group. They had advanced MRI. We were bringing CRISPR expertise. We really were joining forces scientifically in this collaboration. Our part of the collaboration was supported by the Tan Yang Center at MIT, and their part of the collaboration was supported by the Chinese Academy of Science and Shenzhen government and Guangdong government and Ministry of Science and Technology and so on. And by joining all these forces, Guoping Feng was leading the genetic effort from the MIT side, and the current head of the effort on the Chinese side is Zhonghua Liu at SciEd. And we were able to create a group of animals with this SHANK3 mutation. And the results of that first study were published a couple of years ago in Nature. I'll just show you some of the results here. First of all, this is how the animals were created. You use in vitro fertilization and then inject the fertilized egg with this CRISPR construct that will create the mutation, the SHANK3 mutation. It's put into a surrogate mother, and then the offspring can carry the mutation. And then we studied the offspring compared to matched controls. And when we looked at the behavior of the monkey, we saw a number of remarkable things. For one, the monkey showed a lot of these, what we call stereotypies, these very repetitive, odd behaviors. And this has been found in autism as well. And in autism, it varies from individual to individual. It could be hand wringing or hand flapping and so on. But the monkeys also developed their own individual stereotypies. One was a compulsive bar licker, and another was a compulsive flipper, flipping around in the cage, and so on. So we saw these stereotypies. And just like in Phelan-McDermott syndrome, there's a disorder of sleep. We found sleep disorders in the animal. They had trouble getting to sleep. They had trouble staying asleep. There's less efficient sleep. Now, in humans with Phelan-McDermott syndrome, they often have delayed or even absent speech. Our monkeys don't speak, so we can't test that exactly. However, we did notice that they make fewer vocalizations than the normal controls, and that may be related to the speech problem in autism, and that's something that we're following up. We also noticed that they were less sociable, interacted less with other monkeys. And another very interesting thing we found was that if we paired two normal monkeys with each other, they had a certain amount of play, they had a play date and so on. But if we paired one of the normal monkeys with one of these monkeys with a mutation, the normal monkey had less social interaction. It was always like a monkey psychiatrist, and he was picking up on things in the other animals' behavior that maybe even we weren't seeing. So there was an interesting parallel with the social impairments in autism. We saw the impairment on a cognitive task, a set-breaking task, and could be related to the intellectual disability found in Phelan-McDermott. And when we did MRI imaging on the animals, we found differences in connectivity across structures in the brain, both global and local connectivity differences. So with all that in hand, we now have a set of measures that seem to be characteristic of this mutation. And now we're in a position where we could try therapies. Now, of course, drug therapies will be tried before they would go into humans. But we're very interested in whether we could try some forms of gene therapy in these animals, sort of like, again, like was done in SMA. Now, the idea in gene therapy here would be that you take a viral vector, in this case, an adeno-associated virus, and you package the genetic material, and then you inject this into the subject, and that gene will start expressing that protein, making up for the lack of protein because of the SHANK3 mutation. And so, but the trouble here is that this virus that's typically used for gene therapy has a very low payload, very small, and many genes just simply won't fit in, including the SHANK3 gene. So what Guo Pingfeng did was he made a SHANK3 mini gene. It was a version of the SHANK3 gene that seemed to carry out the critical functions, but was much smaller. And when he tried this first, of course, in mice, it looked like it worked. So this is an image from a mouse brain of an animal that had been injected with this virus. And then the virus was carrying not only the SHANK3 gene, but a gene for a fluorescent protein. And so the green here you're seeing is all the expression throughout the brain of the mouse. So it was very promising. And then when the mouse was tested in various measures afterwards, there was a restoration of some of the neuronal and behavioral functions that had been altered in SHANK3. So it's all really promising. And of course, we're very interested now in trying this in monkeys, and this is in progress. But truth be told, this isn't the cure-all for everything by any means. First of all, we don't know this is going to work in the SHANK3 model. And second of all, we're interested in many of these different mutations and all these different forms of brain disorders. And they're all different. They're all going to require different kinds of gene therapy approaches. Just like in cancer, it's not one size fits all. You have to customize it to the nature of the problem. And so what we really need is we need over the next few years, we'll be developing the gene therapy toolbox for brain disorders, taking in a bunch of different techniques that we're working on for vehicles and so on. But one particularly exciting avenue, I believe, for gene therapy in the future is targeting RNA. That was done, for example, in one of the forms of the SMA treatment. And here, Feng Zhang, the faculty member who developed CRISPR I told you about, has been working on RNA editing technologies. And these these editing technologies can be used to manipulate RNA in many ways. Now, RNA is the messenger between the DNA and the protein constructing apparatus in the cell. And you can target the RNA in a way to either correct the mutation of the gene. You can even create new protective alleles that didn't exist before. So for example, there's a form of the ApoE gene that puts you at great risk of developing Alzheimer's disease. But there's another less common form of the ApoE gene that's a protection against Alzheimer's disease, and you could potentially create that. And there's various ways it can be used to modulate gene activity, to modulate the protein function. So there's lots of opportunities there with RNA editing. So now the last thing I want to turn to is another technological approach that I believe is going to be important in treating brain diseases in the future, which is the use of brain imaging. And here I'm going to be talking about the work of John Gabrielli at McGovern Institute, and whose work is being supported by the Poitras Center. And what John is interested in is using brain imaging to predict who will either develop a mental illness or who might benefit from a particular treatment for mental illness. And in this particular experiment, he was trying to identify children at risk for depression. Now, depression is another one of these epidemics in the US, and particularly in teenagers and young people. Many are suffering from depression. We've had many suicides in young people. And it would be great if we could identify kids in advance of their developing depression and then intervene to try to keep them from becoming depressed. So what John did was he studied a group of 10 and 11-year-old kids. One group was at risk for depression because they had at least one parent that was depressed. And the other group was the normal controls. They didn't have a parent with depression. And then what he did was he used some standard forms of brain imaging, testing their reaction to emotional stimuli. And what he did was he measured their brain's response, for example, to looking at happy faces. And what he found was the kids that were at risk of having depression had a smaller brain response in certain structures when they viewed happy faces. But when they viewed fearful faces, they had a larger than normal response, as you might think it might happen. And then he followed these kids when he had these brain data in hand. And he followed them and found that 3 or 4 years later, 40% of the at-risk kids had in fact developed depression. And when he looked back at the brain imaging data, he found that the brain imaging data were better predictors of whether they would develop depression than all the behavioral measures that they had. So it's a very promising way that could potentially lead to interventions, which could be antidepressant therapy, behavioral interventions, and so on. And I think this, and then John is expanding this throughout a whole range of different psychiatric disorders. An even more, let's say, personalized approach to treatment based on brain imaging is being followed by our close colleague Husheng Liu, who's at Harvard and also the head of the company NeuroGalaxy. And Husheng is following up, there have been previous findings showing that if you use transmagnetic stimulation on a particular area in the prefrontal cortex of people with depression, it has an antidepressant effect. But the problem is that if you look at individuals, the human brain is very complicated. It's all these folds and so on, and it's not easy to find the same area exactly in each person. And so, but Ho-Chung has developed all sorts of methods for looking at brain connectivity. And using these connectivity measures, he can identify the same structure in different people, as you see down below. It varies in location a bit. And now, with that information, it can be used to target this area that individualized to the location in that person. And he needs to now going to be starting clinical trials on this in a Chinese collaboration. So I think that gives you an idea of possible avenues that are going to lead us to new therapies in the future, and that future may not be that far away. So I'm going to give you, my prediction of where we're going to be even just 5 years from now. Certainly within 5 years, we're going to have most of the genetic risks for brain disorders. We're going to have new animal models, including primate models, for many of these based on these gene discoveries. We are going to have identified many of the cells and neural circuits that are important in these disorders, whether that's based on genetics or some other aspect of the pathophysiology. And these will become the targets for circuit manipulation and drug development as therapies. We will also be much further along in gene therapies. We're going to be going from proof of concept of animals, and we're going to be moving into clinical trials just in the next few years. And then these new brain imaging modalities are going to help be guiding us towards more personalized medical treatment for brain disorders in the future. Now, I know for Chief, many of you are interested in the entrepreneurial aspect. And here I would say this is a pretty safe prediction, that all of these advances will provide many opportunities to start and work for companies that will turn these academic discoveries into products that will help the public. You got to have a company in between the lab and the university and getting something out there that was going to help people. And many companies have already been started by some of the people that I've already described. I'm involved in a couple joint ventures myself, and there'll be many more opportunities in the future. And if you were interested in finding out more about some of these companies, you can contact me. So lastly, I want to acknowledge some of the support that we've had for doing this work. We've had a lot of support from private individuals and foundations. They've provided the seed funds for novel ideas that are too high risk for government funding. I can tell you that some of these things I told you about were definitely too high risk when they started. They support as much as 40% of the research that we do across our labs right now. They support professorships and fellowships for students and postdocs. 3 of the faculty members I talked about tonight have had chairs established by our private donors. Ed Boyden, Guoping Feng, Feng Zhang have been supported by chairs supported by the Eva Tan professorship and the Jim and Pat Poitras professorships, and that's a great source of support for them. And then lastly, I want to circle back to what I said at the beginning about the importance of international collaboration in these efforts. I think you've seen from some of the things that I've described for you how important this collaboration can be. And I'm going to draw the analogy to the brain. The brain simply doesn't function well unless you have this kind of connectivity across all the different brain structures involved in producing some coordinated output. And I think science has to work the same way. We've got to be cooperating across labs across the world, and this will be the driving force for discovery and the driving force for the new therapies of the future. And with that, thank you for paying attention.
Thank you so much, Professor. That was incredibly inspiring. And for all of us in the audience, again, please put your questions in the Q&A box, or simply like any questions in the Q&A box already that interest you. And our Q&A session will start shortly. So we'll select a few questions with the most likes. And we'll also have a chance for the audience members to unmute themselves. We'll be selecting a few audience members to unmute themselves and ask the questions live. And for now, I'll start with the questions that came in before when the audience members signed up for today's meeting. So the first question, I know Professor probably already talked about this briefly. What are the critical challenges in obtaining data for research in brain and cognitive sciences?
Well, there's often a debate about whether it's money that's holding us back or people. And I'm telling you, it's people. I mean, money is really important, but you gotta have the right people. You gotta have the people with the right talents. And I can tell you how many times, when we're starting a big research project, we're really trying hard to find the right people to drive the science. And this is in the companies I talked about. I mean, that's the #1 challenge for these companies, is getting the right people, and then comes getting the right money. So there you go, it's people. And I hope that many of the MIT students and other young people watching this are going to get into this field, because we need you.
Absolutely, absolutely. And another audience member asked, how will you comment on the future of computational neuroscience?
Yeah, I mean, it's incredibly important. Because I was focusing on some aspects of therapy, I didn't have a chance much to talk about computation. But definitely, as we target the specific neural circuits that are going to have an important effect, we need computational models to help guide our understanding of what's going on in these circuits. And it's desperately needed. Now, and then, of course, it's needed as we develop new treatments. But of course, the other major application of the computational models is going to be in other industries. In AI, for example. I mean, many of us believe that the AI of the future is going to be even more heavily influenced by our understanding of the brain than it has been so far. And so we have a number of faculty members that are really working on that, just at that interface of brain computation and then artificial computation. That's a huge growth field for the future.
Definitely. And the next question is kind of related to the commercial side of the potential commercial side of tech. So one audience member asked, what is your opinion on Elon Musk's Neuralink? And do you think it's aligned with your goal?
Well, I wasn't too impressed with that pig, to tell you the truth. But they have some great technology there. They have something right, which is that if you're going to have these neural interfaces, you want them to be super flexible, and that's something that the faculty member Polina Anakiva was telling you about. That's something that she's worked on. If you try putting a conventional stiff electrode in the brain, just as Polina says, it's like sticking a fork in a bowl of pudding and shaking the bowl of pudding. It's not going to have a good outcome. So you want something that moves with the brain, and that's something that Elon Musk has right. That's the basis of the Neuralink, and I wish them well. Whether that's going to help us in the war between us and the Terminators of the future, I'm not so sure about that. But I think they're working on some good technology.
And the next one is about why is SHANK3 the most popular target among all the risk genes for autism?
Well, we know a lot about it. It affects a lot of people. We already knew from basic research a lot about the SHANK3 protein in the synapse. It's a substantial number of people. So if we have a therapy developed for it, it really will help many thousands of people. And it's a loss of function mutation. So and right now with CRISPR, it's easier to knock a gene out than to put one in. And so it's easier to do. And if you're developing gene therapies, autism is a great place to start, because you're going to see some of the impacts on behavior early in life. You don't want to have to. That's one of the biggest problems with, say, studying Alzheimer's disease. especially in primates, is you got to wait years to start seeing something. So we have some thoughts about that too. But it's it's an extra challenge doing that. So there's lots of reasons for going for SHANK3.
And the next one is about depression. So do you think depression has a physiologic foundation, and do you think that can be cured in the process of brain cognitive?
Well, I mean, the easy thing to say is everything that we do and think has a physiological basis, right? Because it all comes from our brain. I think what most people, when they ask that question, what they really mean is, do you have to treat depression using physiological treatments, and as opposed to, say, behavioral treatments? And depression is one area where the behavioral therapy, particularly cognitive behavioral therapy, has been proven in clinical trials to be effective, as effective as antidepressants, at least up through moderate depression. And the combination of antidepressants and behavioral therapy is especially potent. So, and of course, in all the other brain disorders, behavioral support and so on are absolutely critical. I mean, I don't want to give the impression that all the treatments of the future are going to be biological treatments, because that's not the case. And depression is just a good example of that.
Sounds good. And another person asked, what is the promising direction to alleviate pain and possibly replace opioid?
Well, I talked about that, and from the work of Fan Wang, having identified the group of cells that seem to communicate the critical signal for pain up to the rest of the brain, I mean, that's huge. And I think that that's now zeroed in our attention like a laser, and that we need to think about ways of manipulating that group of cells. And obviously, we want to test this, and we need to test this in people. And that will happen. So I think that that's a really promising area, and that could relieve us of our this dependence on opioids, terribly abused.
For sure. And Winston asked, what are some of the major breakthroughs that you see the future for Alzheimer's and Parkinson's diseases?
Oh, yeah. And in all honesty, I don't have and my crystal ball for Alzheimer's is no better than anybody else's. And I was, one of the many people that were disappointed by some of the failed human clinical trials. And I know the Biogen compound, there's some thought that there's a signal there. I hope there is. But if there's not, we're back to the drawing board. So I can't give you any good predictions on that.
And possibly related to, again, transferring tech out of labs. So what are some of the foreseeable challenges for transferring lab research to venture products?
Well, I have to say that I think that goes pretty well at MIT. There's a very finely tuned process for getting discoveries patented, and then getting them, shopped to companies that want to take these and bring them into products. Sometimes faculty members and other people involved in the research start their own companies to do this. I'd say MIT is one of the most entrepreneurial universities, certainly in the US and even in the world. Now that said, I think there's a parallel entrepreneurial approach in our Chinese institutions as well. And so I think I really don't, I don't see significant barriers to that kind of translation.
Good. And another person asked, what is your opinion on personalized medicine due to the vast heterogeneity in causes of neurological disorders?
Yeah, I mean, that's a great question. I mean, because it's, this is what we're learning in lots of other areas of medicine. The one size fits all, one medicine, you give it to everybody, and so on. And it just doesn't work that way. People are just too different. They have different reactions. Some people are helped, some people not helped at all. And so we need better, certainly the genetic understanding is going to help some, right? Because you're going to do genotyping on everybody, and we're going to have the association between the genotype and the exact mechanism of the disorder. So that will help us, like it's doing in cancer, and it'll be customized. And then we have all, the brain imaging measures and so on are going to help us guide treatment in people in the future. That's going to be a huge shift in the treatment of psychiatric disorders, brain disorders. Is this personalized precision approach, not just one medicine for everybody.
Going back to the question on depression and physiological responses, another person asked a follow-up saying he would like to know the impact of psychosomatic disorders in the brain and the future of research in that field.
Well, by psychosomatic disorders, he means people who imagine that they have disorders, or is he, what the question really is about is that this interaction between the brain and other body organs that together are all part of the constellation of the disorder. And that's huge. I mean, it's attracting a tremendous interest right now. And one example is the relationship between the brain and the gut. And we're finding out so much more about how important our gut is in brain function and the role the brain plays in regulating our gut and in disease and obesity and many things. That's another thing that I mentioned. Polina Anikieva is that she's developed this interest in understanding the gut-brain interaction, and I think we'll be discovering a lot more about that in the future. And that allows for the possibility of doing things in the gut that will help the brain too. So. I completely agree with I think what I believe that question to be.
Yeah, and another person asked to possibly if you could address the trends in stroke research and treatment.
Yeah, I have to say I'm not an expert in stroke, and I know there's been some frustration in progress in stroke, but I'm just not an expert enough to give you really an informed opinion about that.
Yeah, absolutely. And another, I think a mathematics major in the audience asked if there is any intersection between math and neuroscience, and how can we best apply skills in math possibly in the neuroscience field?
Well, that's great. I'm glad that you asked that question, because I'll tell you, when we are evaluating graduate students for our program, the number one thing that people look at is their math background, because so much of what we do now involves, sophisticated mathematical understanding of statistics and so on, the computational models, and we just find that people who have a strong math background tend to make good neuroscientists, and so it's extremely important. Then, of course, you have the whole, interest in understanding intelligence and AI and so on, is all drawing in lots of people with strong math backgrounds into this field. So, yeah, I think it's very, very important.
Awesome, thank you so much. And one last question for today. Professor, do you think human intelligence will happen in the future with brain cognitive science developments?
Do I think that human intelligence will happen? I hope human intelligence will happen.
Human intelligence, my bad.
What's that, human what?
Human super intelligence.
Oh, super intelligence. Well, actually, that's like sort of like the Elon Musk question, right? Because Elon Musk believes that with brain implants, we could improve our intelligence, and that will help us compete against the AI systems of the future, right? I am a little skeptical about that, frankly. I know some people have talked about like code processors for the brain, and like extrasensory perception for the brain through implanted devices and so on. I would be very wary of that, frankly. A lot of the things that people say they want, they want to give our brains the capacity to do, very complex calculations or very, vast expanses of memory and so on. You just look at your phone these days. You know, if you just use your phone, you can do a lot of those same things that people are saying, oh, wouldn't it be great if our brains did this? But the phone's pretty good at it, right? Our smartphones. And they're going to be a lot smarter in the future too. So I think that instead of enhancing our brains through technology, I think our brains will be using external technology that is becoming increasingly powerful. That's what I think.
Perfect. Thank you so much, Professor. And that would conclude our time for today. So we just really want to say a big thank you to Professor Desimone for taking the time today and sharing with us very inspiring findings and insights. And also thank you to all of you in the audience for tuning in. And we very much hope to see you next Thursday, September 10th, also at this time, so 8 to 9 Eastern time. And we'll be hosting episode 3 of our future series, presented by Professor Carlo Ratti, who's the Professor of Practice, Department of Urban Studies and Planning at MIT. So yeah, you can all follow the event updates on our official account on WeChat, Facebook, and LinkedIn. Well, thank you so much again, Professor Desimone. Thank you all for attending, and we very much look to seeing you all next week.
Thanks for having me. Take care.
Thank you. Bye-bye. Yeah, for anyone who's still in the audience, feel free to scan the QR code currently on the screen, and you'll be led to a sign-up page for our next event.